word is used specifically here.
As usual, I don't know what people are saying online, so hopefully
that's it.
Emily
Yeah,
is on that. So is that working? I don't know. No one's telling me. Okay. Okay, so we're going to make a start, and we'll help Jane get a screen shared in after this. So I just want to start by saying that the University of Western Australia acknowledges the Whadjuk people of the Noongar Nation, who are and remain the traditional owners of the land on which it's situated. Whadjuk Noongar remain the spiritual and cultural custodians of their lands, and we acknowledge the strength of their culture and offer our respects to elders past, present and emerging. So today is week 5, and we seem to be getting through some of the issues of the emailing system. So just again to reiterate, please check your junk, spam and or other folders. I have been through the email list myself, and I can absolutely categorically verify that people are on the email list who are still emailing for registration links. So we'll figure that out. I'm going to run a test with Emily Dock about it. So of note as well is that next week's speaker, Pook, she has been called onto field work, and in addition, Sean Winter, who was due to present on the 4th of May, has had to pull out for field work as well. So Pook's talk is shifting down to the 4th of May, and as of today, we don't have anyone lined up for next week. I don't know whether that'll change. You'll certainly be notified in the email at the end of the week. So we record these, but it is up to the presenter preferences and also community permissions whether they are going to go onto our YouTube page. And this also might take some time as well. So, yeah, please recognise that, yeah, weeks, maybe months might actually have to extend before particular talks can go live. And follow us on all of our socials, they're all there. At the end of it all, we're pushing through with just advertising through our Facebook page and then our weekly seminar email list. So it seems to be working. There's a few hiccups still to work through, but we're getting there. So today, joining us, not from too far away, but from Wodjuma, is Jane Skippington. So Jane is an archaeologist and a heritage practitioner with extensive experience, over 15 years. She's currently the Manager of Cultural Heritage on Wodjuma for the Department of Biodiversity, Conservation and Attractions, but has worked extensively in consulting. Here's a great photo I found of Jane. Worked extensively in consulting, and today she's talking to us about her PhD research, which examines shifting human-environment relationships through the isotopic analysis of archaeological macropod bone and tooth enamel carbonates. So she's joining us today. I'm going to stop my share and we will see if we can get her PowerPoint up and working. So welcome, Jane.
Okay, I'm going to start it and see how we go.
Okay, let's see. That's looking good so far.
But it's going to the wrong one.
Oh.
I don't want that. Do you know what I mean? It's showing the wrong side of my screen.
I can see that. So
This way.
Whoops, sorry guys. Some stupid things are open. Me too, Jane. I've got issues here too. Okay, so try that again.
Doing that.
Which?
How many screens have you got?
How many screens have you got? I'm hearing in the background.
So it should tell you which one you want to click. They should give you a number.
Did you hear that?
Yeah.
But where do they do that?
But when you should be able to see, multiple numbers. When you say share media, you'll get a thing that says you can see your. It should come up with all the choices that you can make. There you go. Which is the one you want out of those ones?
Share. Stop sharing. Share.
Have you got your PowerPoint already in slide mode? Start your slide presentation and then select that.
My slide presentation.
Just bear with us, everyone. We've all been here. We can all appreciate the difficulties of technology. So thank you.
It's, yeah, moderately annoying.
Especially for a
Just moderately?
Well, I wish it wasn't like this. But why are they doing that?
So if you share your screen, Jane, we should...
Click the share button and it should give you the choices of what you could be sharing.
This is true.
See the share at the top button, the button next to the leave thingy? See there's a share at the top right? Yep.
Yes.
And it'll give you the choices that you can potentially share with us.
Okay, share.
Oh God. Now we've got chat information.
Yeah,
You have got PowerPoint in presenter mode. Yes. Okay, I think this is screen one. Let's hope this is right now.
Okay.
It's
Looking
not screen two.
Yeah.
Yeah.
So if you just do the little
Click it
thingy
and start the slideshow.
Wrong
Can you?
side. Yeah, but I think I am in the wrong screen. Sorry. I think it is screen.
You're doing now.
We're in the right screen.
It's in the
If you start-
I think
if
it's
you
in the display setting of the PowerPoint.
You do it.
Come on. This has got to be it now. Yes. I think we're there.
Okay, I can see cute doggies. Yes. Yes. Yay! Awesome.
Thank you so much for your help.
You're
academic.
welcome. I'm going to mute my mic and we'll get started.
Okay. All right. So I'll start by acknowledging the Whadjuk Noongar people who are the traditional owners of the land I'm on today. And also I'd like to pay my respect to the Talanji people as traditional owners of Barrow Island. So India's already introduced me. I work mainly on Whadjuk. But I'm also a PhD candidate in archaeology at UWA on the Barrow Island Archaeological Project, and I have been for quite some time, the better part of a decade. But that could change sometime soon because I've recently submitted my thesis for examination, and today I'm going to go through and share just a few highlights from that research. So the fundamental aim of my thesis was to investigate and report on shifting in human environment relationships over the past 50,000 years on Barrow Island through isotopic analysis of modern and archaeological macropod bone and tooth enamel carbonates. And by macropods, I specifically mean kangaroos and wallabies. This aim is or was noteworthy at the time for two main reasons. Firstly, the exceptional significance of Barrow Island as an archaeological landscape, which I discuss more throughout the presentation today. And secondly, the fact that carbonate analysis of Australian marsupial fauna has never been undertaken in an archaeological setting before. So, underlying my overarching goal, there were several interrelated technical and interpretive objectives underlying that. So the first was obviously to do a detailed cross-disciplinary literature review and evaluate the potential and how I might utilise that potential. So that obviously was done quite early on in my PhD. The second aim was to improve the accessibility of isotopic studies on macropod tissue specifically by optimising existing best practices of pre-land analytical procedures for isotopic carbonate analyses. So I'm not going to talk about that in a lot of detail today, which probably people are relieved by because it is quite dense, other than to say that myself and the collaborators I work with have set a particular standard, our own standard for acid pre-treatments of kangaroo bones and wallaby bones. So the two main goals that will be discussed today was to determine the sensitivity of macropod-based isotopic research for detecting intrasite trends and variability across a broad range of contexts, and lastly, to identify and assign those isotopic indications of short and long-term environmental change and interpret their relationship to social and economic activities. So as I said today, I'm just going to focus on highlights. So with that in mind, the principal focus of the presentation is to demonstrate the exceptional significance on largely unfulfilled potential of isotopic analysis by presenting new high-resolution results from 2 main case studies on Barrow Island. Okay, so the first case study examines shifting coastal conditions with respect to precipitation and vegetation from the earliest occupation of Barrow Island at approximately 51,000 BP through to the early abandonment of the island during the Holocene, at the very well-known site of Booty Cave, which you'll probably be familiar with. I imagine people frequently present on that in this forum. The second case study investigates more recent environmental change at a historical settlement site. That site is D24001. This locale served as an incarceration site for interned Aboriginal pearling labourers during the 1880s. Just before I go on, just to set the scene, I want to note that these case studies have been completed in collaboration with my supervisors, Peter Veth, Tina Mann and Alistair Patterson, and they're now documented in co-author publications. So I won't continue to mention exactly who did what all the way through, but thank you very much to my supervisors. All right, let's go. So to start with, I wanted to briefly go through some fundamental isotope theory and briefly discuss its development in archaeology. Some of this stuff will be very obvious to some of the audience, but it's necessary context if you want to engage with the presentation fully. So I thought it was worth investing a tiny bit of time. So in pursuit of just the basics, naturally the most important question is, what is an isotope? Essentially all elements in the periodic table, literally all of them, hydrogen, helium, lithium, beryllium, whatever, all have multiple forms or what are called isotopes. The isotopes are types of the same element. You can see I've got the carbon example. They're types of the same element but differ in abundance and the number of neutrons held within the nucleus. And that difference results in a difference in weight. The difference in weight's really important because this creates subtle chemical differences that influence the cycling and distribution of those isotopes. And that's via different types of reactions, kinetic and exchange reactions. And those processes are broadly referred to as fractionation. So isotopes move around the biosphere and are fractionated by various ecological, physiological processes. So that means they can be used as really powerful tracers for tracking change in ecological systems, which means they can be utilised in archaeology. Just a quick boring note on notation for everyone. The difference in stable isotope relative abundances are often small because of the different distributions in the biosphere. So for that reason, they're generally given in per mil difference in the ratio of the heavier to the lighter isotope. So you can see that there in the equation. So, and it's compared to a standard. So you can see the delta notation is effectively as follows, but as follows on the screen, I mean. But you often hear people say, oh, I want to talk about isotope ratios, but they're usually talking in those delta values, which are in fact a ratio of a ratio. That is the ratio of isotopes in your sample versus the ratio of a standard, if that makes sense. And with that in mind, a negative value doesn't necessarily mean anything in particular other than there's less compared to the standard, not that there's less than 0 isotopes. So isotopic analysis of animal remains in archaeology and paleontology, for that matter, has been around for a really long time. The first zoological, well, a really long time, depending how you think about it, I should say. If you're an archaeologist, you might think it's not that long ago. But the first applications for zooarchaeology and paleontology occurred in the late 70s and 80s. And these were subject to some criticism at first, but over time, the field just genuinely exploded. Isotopic studies today in archaeology have become one of the most applied scientific methods. As you can see on the bar chart, there's a huge number of studies being published every year, like 20,000 in the last decade, which is really quite substantial. There's a few reasons for the explosion. Firstly, just from a technical perspective, accessibility has continued to increase over time. That is, the cost has gone down and processing power has gone up. Secondly, targeted research has resolved a lot of those criticisms of early studies. So at first, people just simply didn't believe that fossil and archaeological samples would preserve a biogenic isotopic signal. But over time, evolution and sort of refinement of pre-analytical processes has occurred, and that's ensured that an accurate signal can be measured. And equally, it's been proven that it can be measured. Finally, as a side note, the theoretical approaches have evolved in archaeology. In a very broad sense, the shift from ecological adaptation models, like those championed by Binford, towards more interpretive post-processual models like Hodder, have allowed for the emergence of complex contextual archaeologies, and these are often supplemented, if not entirely investigated, through scientific methods such as isotopic analysis. So basically, isotopic reconstructions of site histories are really dominant and are an omnipresent feature all over the world, and specifically in Europe, North America and Africa. They're everywhere, or at least a lot of places, but not really Australia. Comparatively, isotopic studies of Australian marsupial fauna are really rare. In fact, the examination of macropod remains in an archaeological context is limited to a single investigation in South Australia that was conducted by Amy Roberts and focused on late Holocene climate change. There have been no studies in macropod biominerals, that is bone or enamel carbonates, which is what we want to do. But why is that the case? Interestingly, it's not due to a lack of foundational research or comparative studies. In fact, the opposite. There's really a rather substantial corpus of foundational literature, which has been led by Brett Murphy and Rebecca Fraser and others. And this literature really quite competently establishes the biological sources of variation. There's also several paleontological fossil studies utilising these relationships, the best known of which is probably Prideaux's 2007 Nature paper, which examined enamel samples from kangaroo species recovered at Thylacoleo caves to reconstruct in past environment. So with that in mind, it makes sense to ask, what are the relationships and can they be utilised in archaeology? Here's what they are. I'll just summarise quickly. So for herbivorous mammals, including kangaroos and wallabies, the isotopic composition of their tissues is primarily a function of diet and water intake. At one level, it's just that simple, but it's also marginally more complicated when you look a little closer. Essentially, the oxygen values reflect the oxygen values of body water, which is primarily influenced by the isotopic composition of ingested water. And it's also influenced by animal physiology. So for non-obligate drinkers, which are like kangaroos and wallabies are non-obligate drinkers, that means they obtain most of their water through dietary plant sources. The oxygen values in their bone and enamel, tooth enamel, reflect the composition of the ingested leaf water they're getting. Leaf water in turn is sensitive to evapotranspiration processes with enrichment of oxygen linked to faster evaporation of the lighter isotope. So it's coming back down to really how fast oxygen's moving through the leaf tissues. It can therefore be used as a proxy for relative humidity. By tissues, I mean cells, apologies. Additionally, the carbon values of animal tissues reflect that of diet. The fundamental basis for the isotopic studies of herbivores is that different photosynthetic pathways of C3 and C4 plants mean that they fractionate the carbon isotopes to different degrees, and thus their contribution to the diet of grazers is isotopically discernible. This can be utilised to estimate diet and therefore reconstruct paleovegetation. So the Barrow Island case studies I'm about to present were specifically designed to test the application of these relationships in a range of archaeological contexts. Just quickly, as an aside, it's important to note that while bone is a frequent feature in archaeological deposits, its isotopic integrity can be easily impacted by diagenetic factors and issues of preservation. Bone carbonates are predicted to preserve for around 1,000 years, which is decent, but not comprehensive. Tooth enamel is far, far more robust. And that's because of its high mineral content and high density crystalline structure. So it's very reasonable and / really necessary to select enamel for Pleistocene contexts. Okay, as I said, the study area is Barrow Island. It is an A-class reserve located 50 kilometres off the northwest coast, as you can see on the map. The local climate is semi-arid and has substantial variation in rainfall. Precipitation is very dynamic and unpredictable, and that's due to the island's position between the southern extent of the modern Australian monsoon and the northern influence of extratropical winter rainfall. Vegetation is primarily C4 grassland, so it's just a spinifex grassland basically, interspersed with the occasional tree, shrub and little herbaceous bits and pieces. There's no C3 grasses on the island, which is convenient, and there's also no permanent free water sources. This limited diversity in vegetation and hydrology is really ideal for isotopic studies, and that's because it limits the sources of variability impacting on the modern samples, and it will reduce the complexity associated with interpreting results. Okay, so the other main reason Barrow Island is particularly attractive for isotopist studies is because of the really significant and intense long-term changes in the environment and climate. During the Pleistocene, Barrow Island would have been joined to the Northwest Cape in a continuous coastal plain. When sea levels were at their lowest during peak LGM, the coastline would have been situated tens of kilometres west of where it is today. The marine transgression that followed cut the islands off from the mainland at approximately 7,400 BP. This slide shows the most recent modelling available for Barrow Island from a pilot study led by Ingrid Ward. It's currently in press. I only just heard about it from Pete, and it's worth looking at. Naturally, precipitation and vegetation would have also really transformed dramatically during this time. Of course, of additional importance is the fact that Barrow Island is overrun with an array of contemporary and archaeological mammals, including macropods. The two species that I focus on in these studies are Euros, also called hill kangaroos, Ospharanta robustus, and spectacle hair wallabies, which are on the right, the Lagorestes conspiculatus. Okay, so case study 1, Booty Cave. The cave, which I'm sure you have heard about before, was subject to substantial excavations as part of the Barrow Island archaeological project from 2013 to 2015, and the ongoing analysis has generated tons of data and has demonstrated exceptionally well preserved cultural assemblages dating from the first occupation of the site 50,000 years ago. This situates Booty Cave amongst some of the earliest known occupation sites with rich faunal assemblages for the continent. This annotated Strat profile from the site, also produced by Ingrid Ward for a joint publication for the project, demonstrates the complex interplay between the rich archaeology and the environmental context. The early appearance of economic and utilitarian shellfish from the end of the LGM continuing throughout the terminal Pleistocene highlights a complex habitation environment and emphasises the requirement for high-resolution climate records. You can see the substantial impact that environmental vectors, including that sea level change, no doubt had on human populations during the site's history. So the pursuit of proximal sources for paleo-environment interpretations, and specifically looking at macropod remains, is strongly justified to complement wider regional models. Really, the considerable dynamism represented implies that, you know, it may well be possible to track change to interstrata resolution, and that was effectively the focus of the study. It seemed to me that you almost certainly could use macropod teeth to track change in Pleistocene sites, but what was the sensitivity for that? So for this study, a total of 69 teeth, mostly archaeological, but also a modern baseline, were examined. The process is to drill whole teeth, separate out the dentine using a micro Dremel with diamond-coated drill bits, and subsequently grinding up the enamel into a fine powder using a mortar and pestle. The powdered tooth enamel are then treated. As I said, there's a specific pre-analytical processing procedure that we developed with hydrogen peroxide first to remove organic matter, and then acetic acid for a specific treatment time to remove any absorbed carbonate. The analyses were conducted at UWA at the Western Australian Biochemistry Centre School of Plant Biology. I also note that I'll show you some statistical visualizations going through. These were conducted in R, and when I'm talking about the statistical significance, that was assessed using a pairwise Wilcoxon rank sum test. Okay, so this is the resulting data. And the question was, can you track environmental change to intrastrata resolution? So I really couldn't looking at this at first. Further analysis was required. But interestingly, this is the way you most often, well, the way I most often see isotopic data presented, which wasn't particularly easy to decipher. So here's a more detailed look, and I'm going to break down the notable trends for you. So the first trend of import, or of interest, I should say, is that a statistically significant difference in the carbon was identified between the two species of macropods that we looked at. The adjusted p-value was very convincing. It was 0.0004 or something like that. The measured enamel applies that the sample taxa subsisted on different mixed diets. So they're both eating C3 and C4, but not in the same proportions. While both of them subsist predominantly on C4, the results suggest that the wallabies, the Lagaresti conspiculatus, incorporate more C3 into their diet. Given that Barrow Island is 70% spinifex or something like that, this is interpreted to represent divergent behavioural and feeding strategies rather than distinct habitats. In particular, the larger kangaroo species, the hill kangaroo, are widely regarded to be preferential grazers, and they intermittently revert to browsing in an arid habitat. But conversely, the wallabies are understood to be selective feeders, and they're a bit more prone to browse. So the other main thing to note on this data, comparison unfortunately did not identify statistically discernible separation between the archaeological and modern teeth, and there were no significant intrastrata differences in this study for carbon. This was true even when a negative 1.2 per mil correction was added to the archaeological specimens, and that was necessary to account for a depletion associated with modern burning of fossil fuels. While there's no statistically significant difference, a number of noteworthy features and trends can be discussed. In particular, for the wallabies, the carbon associated with the island and islandisation phases, i.e. the early Holocene phases, trended more negative than the signals recorded in the layers linked to regression of the shoreline. So that's those at 46 to 36 KAPP. Conversely, the hill kangaroo, the robustus signals, were more positive in the transgression, which was strange, not really, could be strange. That is, basically you can see that there's a reversal in the direction of change between the species. Given that the island and islandisation phases are associated with the climatic period broadly defined by ameliorated conditions, it's likely that the macropods has access to a wide range of lush vegetation, and therefore those opposing tendencies to browse versus graze would have been enhanced. Even when interspecies differences are taken into account, it's clear that the carbon for modern teeth and enamel for more recent archaeological strata are highly variable compared to the early pre-LGM layers. So you can see that you're getting a lot more spread there in SU3. The noisier signal, it's probable that it reflects increased diversity in diet and potentially also vegetation structure. This is likely the result of heightened variation in biological productivity in the arid zone during the Holocene and increases in the rainfall. The oxygen results are far more compelling. The oxygen values are not statistically different for the kangaroos and wallabies, which is what you'd expect. It supports the idea that they're getting similar amounts of hydration from a similar source, i.e. leaves. Given the lack of free water sources on the Barrow Island, like that was reasonably straightforward. Meanwhile, interstratigraphic analysis did identify several significant differences in oxygen, which, as you'll recall, is exactly what we wanted. The stratigraphic units 6 and 7 are both statistically distinct and convincingly so from the modern specimens. So then when you look a bit closer, when the stratigraphic units were analysed by chronological context, i.e. Combined into larger analytical units, further statistically significant differences were identified. In particular, oxygen exhibited statistically significant separation between the modern samples and the strata associated with the pre-LGM regressing shoreline. On the basis of the vast majority of the physiological and environmental factors that may have influenced that signal are negated by the island environment, this difference is interpreted to reflect a genuine change in relative humidity. Broadly, the higher oxygen associated with late Pleistocene samples implies dry conditions, and while contradicting some regional models, some existing regional models that suggest that marine isotope stage 3 was wetter than present, it confirms a more recent syntheses by De Decker and Veth and others. So it's basically aligned with what you would expect for that area. In addition to that, the Holocene oxygen was significantly more negative than the late Pleistocene samples. Given that many of the Holocene samples are derived from stratigraphic unit 3, it's probable that this result aligns with the earliest Holocene low identified in the regional Cape Rain stalagmite record. So yeah, that was good. Did it work? Yes, it did. Was it worth it? Yes, it was. When you're doing these kind of studies, you spend a lot of time drilling and looking at teeth with very little idea what you're going to get out. So it was really great to see those statistical differences pop up. Overall, the paleo-environmental signatures in the archaeological teeth from Booty Cave really do track shifting environmental conditions and to a lesser extent evolving habitats. This study fundamentally demonstrates for the first time that isotopic analysis of macropod tooth enamel can be used to track paleo-environmental shifts and can do so to interstrata resolution. So that was a good result. You can produce useful data in Pleistocene contexts, which of course begged the question, what else might you be able to do? So that's the next case study. For this case study, I'm going to dramatically sort of shift scale to examine recent change at a historical site. We've also shifted direction. We are now south of Booty Cave at the Bandicoot Bay Settlement. As most of you know, the settlement and colonisation of Western Australia by the British was initially restricted to the southwest, but by the mid-1800s, commercial opportunity linked to pastoralism and extractive industries materially expanded activities into the northwest Australia. During the late 1880s, it was pearling fisheries that peaked as Western Australia's most critical colonial export. So sadly, historical documents suggest that thousands of Aboriginal labourers were caught up in this and subjected to deplorable maltreatment and translocated large distances from their homes to support the pearling industry and a practice referred to as blackbirding. This included marooning labourers on remote offshore islands in periods of hiatus from their programs. So importantly, the Bandicoot Bay Settlement site preserves rare evidence for this type of geographic incarceration and therefore a unique opportunity to consider Aboriginal agency and adaptation in the face of coercive colonial practices. The site is in a shallow but expansive sand dune, you can see on the slide, and it's located in a low relief sand dune. Okay. So an open area excavation was conducted, led by Alistair Patterson, between 2013 and 2014. And there's been quite a lot of analysis done by Tom Dooley, Tina Mann, Shay Byrne, others. The assemblages suggest that the subsistence strategies employed by internees mirrored the approaches of mainland Aboriginal communities and focused on the procurement of and processing of proteins, particularly the local wallaby. derived from nearby habitats. So critically, the reconstruction of past environments through isotopic analysis of those wallaby remains at the site has reasonable potential to provide further insights into the strategies employed by internees to manage what were undoubtedly really harsh conditions. So here we go. Again, to investigate this, we used bone rather than enamel, and the isotopic analysis was conducted at UWA. The pre-analytical and analytical processing of the samples is really broadly similar to the enamel samples that I presented earlier, so I won't go through that again. So this was the output from the bone samples. You can see there's a really clear pattern here. We got a very convincing, statistically significant differentiation between modern and archaeological samples for both the carbon and the oxygen. So what does that mean? Firstly, a statistically significant shift was observed between the modern and archaeological oxygen. This result is interpreted to reflect a change in relative humidity. When I extrapolated this from other data sources, I suggested that it was around a 3% change in that humidity. There's not very good historical rainfall data for Barrow Island. However, state records from the early 1900s through to contemporary observations indicate that the magnitude and direction of long-term trends in rainfall vary across Western Australia. The northwest has seen an overarching trend towards increased summer rainfall, but rainfall precipitation trends do decrease in the Pilbara, for example, from the 60s to around 2012. So it's not straightforward. Considering the relatively short period of time being examined by this study, combined with the climatic dynamism of the region, it's possible that the shift in oxygen values reflects a change in the local weather conditions. It's really plausible that the more positive archaeological isotope signatures relate to drought conditions experienced at the continental scale in the mid to late 1880s. And those wide-scale droughts are reported in various newspapers at the time. Although the change in relative humidity is relatively small, it would represent a really significant reduction in an already arid landscape. Okay, now the carbon. Again, a statistically significant separation was identified, and this notable difference is evident even when the correction is applied that I described before for fossil fuel burning. It is possible that the significant shift reflects variation in water availability, or alternatively, it may be linked to a shift in vegetation resulting from a change in local conditions. I used a two-source mixing model to approximate and compare the relative contributions of C3 and C4 plants between the two contexts. In this case, a maximum archaeological dietary signature of, like a dietary component, I should say, of C3 is estimated to be around 40%, whereas the modern dietary C3 contribution is estimated to be higher at around 50%. So that is, the archaeological wallabies were eating a less diverse diet made up of a lot of spinifex compared to the modern samples. Given the oxygen results suggest that the historical occupation likely intersected a period of drought, it's possible that the lower contribution of the C3 plants could reflect a significant decrease in diversity of vegetation linked to changes in precipitation. However, this seems somewhat unlikely based on the small magnitude of change implied by the values. Equally, the more positive carbon results for the archaeological samples are not thought to reflect a behavioural change. That's because the non-obligate drinkers are prone to browse. They'd favour moisture-rich C3 plants over C4 in response to drought conditions. So it's the opposite of what you'd expect if that would be the case. Instead, the change may be linked to a sharp change in vegetation, and this could be related to a significant event such as historical anthropogenic burning and or lightning-induced fire. Such an event has the potential to result in an intense short-term grazing of the reshooting C4 grasses, and that would explain the decreased reliance on C3 plants. Earlier historical burning on the island occurred in 1863 as part of the landing party. The island was fired from end to end. And it's not improbable that incarcerated pearling labourers also undertook cultural firing activities, perhaps to clear the site or perhaps to facilitate macropod hunting. Like case study 1, the historical research has provided an interesting result. The isotopic and archaeological evidence for the marooning of Aboriginal internees on a remote island characterised by a lack of free water sources in a time of drought demonstrates the shocking extent of exploitative practices utilised by the pearling fisheries. The arising necessity for Aboriginal people to potentially utilise traditional knowledge for survival undoubtedly provided an important economic opportunity for the pearling operations to relieve strain on their provisions, but also in the context of the trauma likely experienced by the internees, the short-term requirement to jointly re-engage in cultural activities and use cultural knowledge may have provided an opportunity for Aboriginal people to reconnect with group identity and signal resistance for that matter. So I'd just like to conclude with a few final remarks that emphasise what was achieved by these studies relative to the initial objectives, of course. So overall, the case study sought to investigate the sensitivity, efficacy and usefulness of macropod-based studies in a range of contexts. Results from Pleistocene and Holocene age tooth enamel results track dynamic transformations in the local environment over time. This includes the identification of clear shifts in oxygen values indicative of the onset of aridity leading into the LGM and a period of increased humidity in the early to mid-Holocene. The enamel carbon results demonstrate divergent feeding strategies and imply, perhaps a shift towards increasing diversity in vegetation structure. That's good. This is evidence for considerable environmental change through time, and it starts to shed light on the complexity of early coastal occupation. The research has also confirmed the relevance of macropod analyses for identifying more recent short-term environmental change through investigations conducted at the historical site, which were perhaps, it was an eye-opener, I would say. I wasn't too sure that would be possible. Oxygen values and historical climate data indicate occupation of the site likely coincided with a period of drought, and this highlights the difficult conditions experienced by internees. The carbon values are interpreted to reflect variation in water availability, or possibly that short-term vegetation change we discussed. So yeah, essentially it works, but it's not straightforward. Considerations need to be made as to whether it's appropriate. Barrow Island really is the optimal setting for this kind of isotopic work. That's it.
Thank you, Jane. That was a very impressive presentation. Incredible amount of research there.
Thank you. Just stop sharing my screen, eh? No, no how.
Okay, so turning to discussion. Oh, actually, I can probably turn my computer this way. Are there any pressing questions from the room? Jo, we have Jo, and you should be able to hear her from there.
I'll come here, just in case. Hi, that was great, Jane. Thank you so much. I'm interested in the possibility that your internees living on Barrow Island, in that sort of prison-like period of pearling, could they have actually changed the vegetation enough to have got rid of all the variety that in fact you're finding in the historic period? So in fact, may it have been a drought, but may it also have been the landscape that was in fact being managed by humans who are actually there burning spinifex and doing various different things and in fact changing the landscape that way?
I mean, hypothetically, yes. I thought about it. It's possible. It is possible. When I think about it, I suspect if the site was burned, if there was a sharp vegetation change there, I do think it was for clearing the site rather than managing the landscape. Because it's very concentrated around the site. So it's literally the modern specimens and the archaeological specimens come from that direct area. It's really just tracking what's happened right there. So it doesn't, at this stage, really capture what was possibly happening on the whole island. And that's a much bigger undertaking, I believe, because we need to do sampling across all the different vegetation units. Even though it's just a simple spinifex grassland there are kind of micro vegetation areas, if that makes sense.
Yeah, so when people were left in the barracoons, were they locked up while the pearlers went away, or could they roam across the entire island, or were they in fact somehow constrained to the booty cave? Maybe that's a question for Al as much as for you.
Yes, the island was a prison, but I think it's interesting to think about what you're saying, Jane, as well, because the modern Barrow Island, like many modern environments, is kind of unnatural in terms of archaeological time. So the spinifex there is huge, and it's basically been, fire suppression is in place for the last 50 years through oil and gas. So I do think what your results are great, because I think they actually show us also how different the modern condition is, and the samples that you're getting are from even, you know, that kind of late 1800s inside, because this island hasn't been used for 7,000 years. So, you know, anthropogenic burning isn't in place. So really it's only in the 1800s we have a bit of burning and then it stops. So it's a really interesting moment that your sample picks up on. And so that's why there's such exciting results. So.
Yeah.
Thanks.
But yeah, Joe, I guess, like, to answer your other question, they definitely must have been able to roam, because they couldn't have survived at that, just in that locale with no water.
Yeah. Yeah.
Have we got any questions in the chat, Em? Or
No, not yet.
no?
There's lots of congrats. Oh, yes, we have.
Oh, hello.
Can you see that or here?
It would be great if you could read it out.
So Piers has said, thanks, Jane, very interesting. What are the main uncertainties you identified in the isotopic data, if any? E.g., might diagenesis have an effect? Are there any possible issues regarding the age of the marsupial teeth sample, as in like life cycle stage? And what might be needed to further develop this sort of method? So like 15 questions in one.
Yeah, got it. So diagenesis, I feel really confident that the Pleistocene samples are great. I think the pre-analytical processing is effective. But more than that, I'm not entirely sure it was necessary. I think the enamel was reasonably preserved over that time period. So that's good, even in a limestone cave. I think the bone is more of an issue. Ideally, I'd like to do other work, you know, RMS or something, to confirm that there was no change in that historical bone. I don't think there was, because we did pre-treat it, and you know, it's a really constrained environment, it's a really short time, but you can never know for sure, and the results are just so clean. Like, the statistical significance is so convincing. It would be really great to potentially test that, so you can be 100% sure, but I'm reasonably confident. In terms of the life cycle of mammals, and particularly wallabies and macropods, they do have a weaning effect that's recorded in carbon. So when they're feeding, when they're being fed, their mother sits at a different trophic level, or, you know, however food webs work. So there is a weaning effect in the carbon isotopes for young animals. But with macropods, you can tell by when the teeth erupt. So the later stage teeth, the molars 3 and 4, are targeted so that you're not getting that weaning effect. I did do some experiments where I looked at jaws to confirm that weaning effect, and it does appear to be fairly solid. So yeah, to conclude, you really need to focus on teeth in adult specimens. You don't want to include young animals. I can't remember the last part of the question. What was the last bit? Sorry.
But what might be needed to further develop your method?
In an ideal world, you do a really dense, huge number of specimens for the modern samples. So we could look over the whole island and construct variability for the island. And again, I think it would be great to do more testing to look at potential diagenesis. Like there'd be other methods to pick up any intrusive carbonates. I don't know the extent to which is necessary for these case studies though.
Fantastic, thank you.
Do you see that as part of rolling forward with this, Jane? You know, just more research. Do you think it'll be uptaken or uptook by the generally more by the archaeological community, that isotopic analysis will become a bit more of a norm here in Australia?
I really think it could, because the paleontological work that's been done is really convincing and interesting. So you are getting a lot of paleontological sites that have a reasonable number of samples. I guess the limit for Australian archaeology, well, one of the limits, is that not all sites have intact jaw bones. Not all sites have a reasonable preservation of bone. So I think it could be taken up by some people in some sites. It's reasonably straightforward in the method sense. Once you have the samples, it's a matter of processing them and analysing them. So in that way, and it's not too expensive really in the scheme of things either. So it is attractive, but the sampling and the sites you target would always be the limit.
There's a comment I just saw pop up from peers. Any thoughts of applying it to rock nest?
Well, we don't have any sites that I can think of to target. No, I don't usually. I mean, obviously I work on rock nest, but the sensitivities around doing any kind of destructive analysis on an incarceration site of that nature, for people who don't know, it ran as an Aboriginal prison from 1831 through to, well, 1838 through to 1931, and it's still a source of extreme trauma for people. So archaeological works are complicated. You probably are aware that we're starting to talk to UWA about, you know, establishing an archaeological presence again there, but it's really slow going. I wouldn't mind having a look at all the modern quokkas to see how much variation there is in their diet from the settlement versus, you know, West End remote areas. But that's obviously not an archaeological question. It'd just be really interesting to see how much diversity there is in their isotopic selves.
For sure. Any other questions from the room? No. Well, that was great, Jane. If there's nothing else coming through on the chat, which I'll just check. No, I think we've covered all of that. Just on a final note, I was interested to ask, how does community feel about these new results, especially in terms of the fact that it's got such a recent, incredibly traumatic period? How does the Aboriginal groups that you've worked with on BAP and through this research, how have they received this research, you know, elements of your results of the research?
To be honest, we've only just finished this, so it hasn't been widely shared. And I don't know, Al might be able to talk a bit more about it potentially, I don't know, because it mirrors the results from the zooarchaeological remains and also the anthropological remains. This idea that the site was quite painful, sort of well known.
It's only just been published, so we haven't really had much. It'd be nice to see what kind of uptick it gets. But I think when the thing about Bandicoot Bay is it's like, you know, Shay's work, your work, Tom's work, it's really hammered a lot of kind of archaeozoology and scientific analyses at a site like that. So I think you know, it'd be interesting to see how many people take that. It's published kind of in international journals, so maybe, you know, over the next 6 months we'll be tracking that a bit.
I can speak briefly to that.
Pete's coming.
The final, if you like, preliminary results have been shared with Calendly, and indeed they're really quite centred on the fact that there was this very profound period of incarceration, and so they're scheduling a major board meeting to have a presentation of this final data. People are talking about it right throughout the Pilbara, and it's kind of acting as a bit of a stimulus to have more communities from Shark Bay all the way through to Bidgie at Grange to talk about the pearling days. And they do discriminate between the early unregulated period and the later pearling, where it was more, you know, traditional and where people had some agency. But this first phase is seen as highly brutal. And it is, as Al and I described, a form of slavery, which really has just got to the public domain for the first time.
Oh, very traumatic note to end on. Sorry, guys. Sorry for that. Well, I think, sorry. I think that's it, and we'll say thanks to Jane. So thanks to Jane.
I don't get in enough.
Yeah, please come and visit. Well, and I've just got an update which will go out in the email at the end of the week. So we've got Jo Mack is stepping into next week's seminar, and she's going to be presenting on whale feasting and whale watching, how the rock art of Sydney and Murrujuga reveal a different human-animal relationship with charismatic megafauna.
Wow,
So,
that sounds good.
So that's up for next week, and yeah, we'll sign off from here. Thanks, everybody.
Thanks, Jane.
Thanks, Jane. That was awesome. Oh, sorry, I'm not putting my computer.
So, where am I?
You're a while off, Al.